Now, let's talk about the role of the endocrine system in the aging process. The endocrine system is a collection of glands that secrete hormones directly into the circulatory system. There, these hormones travel directly to their target tissues and can thus affect large-scale changes of the entire organism. Not surprisingly, therefore, the endocrine system regulates some of the most important aspects of animal development, metabolism, and maturation. For example, endocrine hormones control transitions between different developmental stages and are responsible for sexual maturation. They regulate overall metabolism, including energy storage and usage, and they even regulate the stress response. Its major glands are the pituitary and pineal gland, the thyroid, the adrenal gland, and the pancreas, as well as the sex-specific testes and ovaries. On an organismal level, these glands are highly interconnected and often operate via feedback loops. The thyroid system, for example, consists of the hypothalamus, especially the pituitary gland, and the thyroid glands, and thus is referred to as the hypothalamic pituitary thyroid axis, or HPT axis. The hypothalamus monitors circulating levels of thyroid hormone, or triiodothyronine, T3, and thyroxine, T4. When T3 and T4 levels are low, it responds by releasing thyrotropin releasing hormone, or TRH. The TRH in turn stimulates the pituitary to produce thyroid stimulating hormone, or TSH. The TSH in turn stimulates the thyroid to produce thyroid hormone until levels in the blood return back to normal. The whole system is connected via feedback loops so that T3 and T4 shut down the stimulating function of the hypothalamus and the pituitary. The function of the thyroid hormones is to increase basal metabolic rate, including fat, sugar, and protein metabolism. They thus act on every cell in the body. They also affect developmental processes, including bone growth, as well as internal heat generation. Finally, thyroid hormones make cells more susceptible to the function of catecholamines, or stress hormones. Thus, the thyroid system is closely linked to the stress system, which consists of the HPA axis, or hypothalamic pituitary adrenal axis. The hypothalamus can release CRH, or corticotropin releasing hormone, and vasopressin in response to stress, physical activity, illness, cortisol blood levels, or is even affected by the sleep-wake cycle. These two hormones in turn stimulate the pituitary to release adrenocorticotropic hormone, or ACTH. ACTH in turn acts on the adrenal cortex, which produces glucocorticoid hormones, which in humans is mostly cortisol. Once more, a negative feedback loop exists, as glucocorticoids in turn suppress CRH and ACTH production by the hypothalamus and pituitary. Glucocorticoids are the body's major stress hormones and can affect sugar and protein metabolism, the immune system, as well as the inflammatory response. One interesting target of glucocorticoid action is the increased production of epinephrine, also known as adrenaline, which is the fight-or-flight hormone in the body. Epinephrine belongs to a class of molecules called catecholamines, the action of which can be potentiated by the activity of the HPT axis, as we've just discussed. The hypothalamic pituitary gonadal axis, or HPG axis, refers to the interplay of, well, you guessed it, the hypothalamus and the pituitary with the gonadal endocrine glands, the ovaries in females and the testes in males. This axis controls development and reproduction. Gonadotropin releasing hormone, or GnRH, is secreted, just like all the other hormones, from the hypothalamus, which in turn stimulates, once again, the pituitary glands, but this time to produce luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH, which in turn act on the gonads to produce estrogen and testosterone, respectively. Once again, negative feedback loops exist to turn off this signaling pathway. These three pathways are also often collectively referred to as the neuroendocrine system, as they directly involve neuronal systems within the hypothalamus and the pituitary. Another important endocrine system is the RAS system, the renin-angiotensin-aldosterone system, which regulates fluid balance in the circulatory systems and as such affects kidney or renal and blood vessel or angio function. Blood pressure loss due to bleeding or dehydration is detected in the carotid sinus, a blood vessel near the heart that is equipped with highly sensitive pressure receptors. In response, the kidney will now release the enzyme renin. Renin acts by cleaving a protein called angiotensinogen into its active form, called angiotensin 1. Angiotensin 1 is converted by angiotensin 1 converting enzyme, or ACE, into angiotensin 2, which is the final active compound of the system. Its activity include vasoconstriction, or tightening of the blood vessels to increase blood pressure, and increases water and sodium reabsorption rates within the kidney to aid in fluid retention. You may have heard of ACE inhibitors, which are used to treat high blood pressure, as they inhibit the activity of ACE. Thus, no angiotensin 2 may be produced, and blood pressure can be lowered. Finally, there is the HPS axis, the somatotrophic axis, technically also part of the neuroendocrine system as it involves signals from the hypothalamus and the pituitary. This signaling network regulates growth and energy metabolism. The hypothalamus responds to many physiological stimulators, for example, exercise, nutrition, or sleep, or inhibitors, for example, free fatty acids, with the secretion of either growth hormone-releasing hormone, GHRH, or somatocrinin, or growth hormone-inhibiting hormone, or GHIH, or somatostatin, which in turn stimulate the pituitary to secrete growth hormone, or GH. GH itself is a major anabolic hormone that leads to the buildup of muscle mass and bone mass. In order to acquire all the energy required for this building activity, GH also leads to increased fat breakdown and the release of free fatty acids, which in turn can be broken down into energy. This system is tightly connected to the hormone ghrelin, also known as the hunger hormone, which is secreted by the stomach when the stomach is empty. Ghrelin regulates the feeding response, in which it is opposed by leptin, the satiety hormone, which is made by the adipose tissue. Growth hormone furthermore acts on the liver to stimulate production of IGF-1, or insulin-like growth factor 1, which is one of the most important growth factors of the body, as it stimulates cellular growth of almost every single cell in the body. IGF-1 is related to insulin, a hormone released by the pancreas in response to food intake. The role of insulin is to induce cells to take up glucose from the bloodstream. After a meal, glucose levels in the blood rise as nutrients get absorbed by the intestines. Type 1 diabetes, the inability of the pancreas to produce insulin, and type 2 diabetes, the inability of cells to properly respond to insulin, result in improperly controlled blood glucose levels. Elevated blood glucose levels have detrimental health consequences, as they can damage cells and macromolecules alike, especially so-called advanced glycation end products, or AGEs or AGEs, which are the reaction products of the reaction of glucose with especially proteins, have drawn the attention of researchers and physicians. These AGEs represent molecular damage, and this damage may in turn result in tissue and organ failures, or even premature death. With endocrine pathways involved in the wholesale regulation of so many different body systems, it is not surprising that the very first longevity gene that researchers identified, called AGE1, was identified in a molecule that is involved in mediating the effects of endocrine hormones. Specifically, AGE1 is involved in the cellular response to insulin-like growth factors in a signaling pathway called insulin/insulin-like growth factor signaling, or also abbreviated as IIS. The IIS is conserved in diverse animals, from invertebrates, such as nematodes or fruit flies, to mammals, such as humans. Details differ, though. For example, C. elegans has more than 30 insulin-like hormones, called ILPs, for insulin-like peptides, and fruit flies have at least 7 ILPs, while humans have much fewer. We have insulin, and insulin like growth factor 1 or IGF-1 and IGF-2. But in contrast, the effects of these ILPs in vertebrates are mediated through a single receptor for ILP called DAF2 in worms or INR for insulin receptor in flies. Mammals have a variety of receptors for ILPs, including an insulin receptor, INR, and IGF receptors. Nonetheless, the basic signaling mechanisms are essentially the same in all these species. Signaling starts when the ILP ligand binds and activates its specific receptor. This in turn activates a cascade of proteins, including a class of enzyme called PI3 kinases or PI3Ks. In worms, this PI3K gene is called H1. As mentioned before, the very first longevity gene that was ever discovered. PI3K activation in turn activates a protein called ACT, sometimes also called protein kinase B, which in turn can inhibit a transcription factor of the FOXO class of transcription factor. In worms, they're called DAF16. A transcription factor is a protein that can bind DNA and regulate the expression levels of genes, either by promoting their expression or inhibiting it. Activated FOXO/DAF16 moves into the nucleus, finds specific sites on DNA, and modulates expression of genes. Activated FOXO, or DAF16 in worms, moves into the nucleus, finds specific sites on DNA, and modulates the genes associated with these sites. ACT activity on FOXO prevents that movement and keeps this transcription factor sequestered in the cytosol. Many more proteins are involved in these pathways, but the principal players are the ones listed in this figure. In C. elegans, the IAS interfaces with another hormonal signaling pathway, this time involving a protein called DAF12. DAF12 belongs to a class of transcription factors called nuclear hormone receptors, or NHRs. When a ligand binds to these receptors, they go directly to the nucleus to regulate gene expression, without the need for a complicated intervening signaling cascade. Ligands for NHR are usually highly lipophilic substances, such as cholesterol or derivatives of cholesterol. DAF12 ligands are called dafachronic acids and resemble in their molecular structures the bile acids found in mammals. Interestingly, synthesis of these dafachronic acids depends on an enzyme of the P450 class of enzymes, which in turn can get activated by IIS. The nematode homologs of these genes are called DAF something or other, due to their role in Dauer formation. Dauer is a German word that means enduring. Hence, the Dauer state in C. elegans is a state that the developing animal can enter before it reaches full maturation in adulthood, when environmental conditions are poor or unfavorable. For example, if food supply is low. C. elegans larvae in the Dauer state are extremely stress resistant and can persist for very long periods of time. When environmental conditions improve, for example, food availability improves, which also means more available cholesterol, both the DAF12 and the DAF2, DAF16 pathways get activated, the worm exits Dauer state, and resumes normal development and reproduction. Thus, IIS and also the DAF12 pathway are growth and developmental pathways important for the timing of growth processes in response to environmental stimuli and environmental conditions. However, in adult animals, they also regulate longevity, as it is sufficient to disable IAS signaling in adults only to obtain long-lived worms. Some of the most robust and most reproducible effects on longevity are in fact obtained using IAS mutants. In fruit flies, the IIS works in very similar ways, where a single INR receptor is responsible for the action of several different ILPs. The initiated signaling cascade similarly involves PI3K, ACT, and a FOXO transcription factor. One peculiarity of the fly system is, however, that the cells that secrete the ILPs, called insulin-producing cells or IPC, are located within the fly brain in 2 clusters of 7 cells each. In other words, Drosophila have their pancreas in the brain. As in worms, inhibition of IAS leads to long-lived animals. This was shown in animals that lack INR, that lack an adapted protein called Chico, or had permanently activated FOXO. Even flies without certain specific ILPs, or even flies without IPCs, are long-lived. However, nothing in biology works in isolation, and neither does the IIS. Flies were instrumental in deciphering the interaction of the IIS with another important signaling pathway in the regulation of aging. This other pathway, named after its key protein TOR, or target of rapamycin, is an amino acid responsive pathway that controls the rate of protein synthesis. We will talk more about TOR signaling in a later module on protein homeostasis, but for now, IAS and TOR interface at many levels. For example, FOXO controls the transcription of a gene called 4EBP, short for elongation factor 4 binding protein. As the name implies, the 4E binding protein binds and inhibits the elongation factor, or E4, which is a key factor in protein synthesis. This leads to reduced protein synthesis. Due to this discovery, the TOR pathway was investigated for its ability to modulate lifespan. Inhibition of this pathway, either via genetic manipulation or by using the drug rapamycin, does indeed impact longevity. It is unclear at this point whether these two longevity pathways are completely independent of each other, completely dependent on each other, or a mixture of those two. The most likely scenario is that since they share some molecular targets, they work in tandem to mediate certain aspects of aging and longevity, while other aging phenotypes are mediated by one pathway and not the other, and vice versa. Modulation of neuroendocrine signaling has even been shown in mammals to affect longevity. In fact, hormonal signaling mutants with altered lifespans were known for a long time before their importance to aging was really well understood, and molecular aging research had taken off. So-called Ames and Snell dwarf mice have lowered body size but are exceptionally long-lived. While carrying different genetic mutations, both Ames and Snell mice lack growth hormone, thyroid-stimulating hormone, and prolactin. Interestingly, Ames mice treated with GH had their longevity restored almost back to normal, highlighting the importance of neuroendocrine signaling, including GH signaling, in longevity determination. The role of GH in aging and longevity is further underscored in a model of GH deficiency, the Laron syndrome mouse, which is a natural mutation in the GH receptor gene, or also shown in the targeted growth hormone receptor knockout, or GHRKO mouse, that was created in a laboratory. One caveat in the analysis of these mouse models, however, is that these animals have genetic deficiencies. That is, genes are functionally disabled, which means all processes that depend on these genes, including developmental processes, are affected. Hence the dwarf phenotypes, which is a growth phenotype that happens during development. Many researchers therefore think that the observed longevity phenotype may be less the result of delayed aging per se, but probably more attributable to altered development. Once again, aging and longevity cannot really be used as synonyms, and a lot hinges on the definition of aging. While degradation of body function essentially starts with birth, as we have seen earlier, aging is usually only considered to set in once all developmental processes have stopped, once adulthood is reached. Regardless, all these GH deficient mouse models also show a delay in age-associated diseases, suggesting that at least a part of their exceptional longevity is indeed due to delayed aging. Since GH is the key stimulator for the production of IGF-1, primarily from the liver, as we have discussed earlier, and to an extent also controls the levels of insulin itself, it stands to reason that insulin and IGF-1 are directly involved in mediating these longevity effects in mice. In fact, Ames and Snell mice have reduced IGF-1 and insulin levels. Thus, many different mouse models for reduced IGF-1 signaling were created including tissue-specific experiments in which the IGF-1 receptor was only deleted in very specific tissues. Many other mouse models have been established with disruption or attenuation of IGF signaling. Common to all of these models is a dramatic increase in longevity, further underscoring the importance of hypothalamic, pituitary, somatotrophic signaling for lifespan modulation, especially in mammals, or IIS in general. Why would reduction in IIS lead to extended lifespans? Data, especially from the mouse models, but also from fly and worm experiments, suggest that reduction in somatotropic signaling increases stress resistance. Cells derived from these animals survive paraquat treatment much better than wild-type cells and have higher levels of antioxidant enzymes. Worms and flies directly exposed to paraquat, which induces oxidative stress, as we discussed earlier, further live longer if they have reduction in IIS. If somatotropic signaling is reduced upstream of an INR or IGF receptor, these animals furthermore have improved insulin sensitivity, that is, their cells respond faster to smaller amounts of insulin and are thus able to clear glucose from the circulation much faster. That, in turn, reduces the toxic effects of glucose. Finally, since IIS plays an important role in cell growth, reduced activity of this pathway also significantly reduces cancer incidence, which is, of course, the unregulated growth of cells. This makes sense, as it is an absence of signals that would stimulate a cell to grow uncontrollably. Data from human studies paint a less clear-cut, slightly more complex picture. Reduction of insulin signaling lies at the heart of diabetic health problems, while reduction in growth hormone and IGF signaling affects growth, and more importantly, cardiovascular health. However, especially centenarian populations are enriched for individuals with lower circulating IGF levels and or mutations in other components of the IIS. Interestingly, this is often associated with shorter stature. The reason for the inconsistencies in experimental results are not quite clear. A very probable explanation is that IIS in humans is simply more complex and has many more interconnected moving parts, such as a different array of ligands, of hormones, and receptors. In addition, there exists a higher level of crosstalk or interaction with other signaling pathways. In addition to the TOR pathway, for example, the IIS also connects to the MAPK pathway, which is a major pro-mitotic and pro-growth pathway. It could be that the traditional IIS is responsible for stress and aging phenotypes, and the MAPK arm is responsible for growth phenotypes. Depending on where along these pathways a mutation occurs, different health and aging phenotypes may thus be the consequence. Check your knowledge. Compare and contrast the different endocrine axes. Explain the concept of molecular signaling pathways. Describe the components of the IIS. Explain the consequence of lowering somatotropic signaling in worms, flies, mice, and humans.